Wait, Dr. Preeti, because still not started live over there.
Yes,
Yeah.
sir. You can start now.
Start.
Okay. So, good evening, doctors, and good evening, all our faculty members and delegates for this beautiful evening where it is a pulse of experience webinar. This is been organized by Cadila. As you know, we as an organization is 70 years old pharmaceutical organization and having many products and therapy in last 7 decades, which is first time in India, first time in world. And with this connection, in fact, I am Dr. Devesh Das, President of Cadila. Do welcome all of you in this beautiful evening of knowledge sharing and exchanging platform. Today, we have with us Dr. Yatin Mehta, who is of Vedanta Institute of Critical Care and Anesthesiology, Vedanta Medicity, Gurgaon. Dr. Rajat Agrawal, who is the Director and HOD of Critical Care, Fortis Escort Heart Institute, New Delhi.
Oh.
We have Dr. Arindam Kar, who is the Additional Director of Critical Care, Sir H.N. Reliance Foundation Hospital, Mumbai. And Dr. Hiten Karelia, who is a Consultant ID, Director, Prime Infectious Care, Vadodara, Gujarat. So I welcome all our distinguished speakers and faculty for today's program. And I also welcome our medical advisor from Cadila, Dr. Preeti, who will be also a part of this today's program as a co-moderator and from our side. I also welcome our all Cadilians, along with all the doctors across the country who are attending the program. So today we have the delegates, especially from physician, medicine specialist, intensivist, IDs, chest specialist, and some specialist surgeons are also there from across India as well as Nepal. So I welcome our Nepal doctors also, because Nepal we had a very good scientific program last month along with the KVLs and KBLs. So before I hand over the session to our distinguished chairman, Dr. Yatin Mehta, I'll give a small brief introduction of our chairman. Dr. Yatin Mehta, as I have told, he's a chairman, Vedanta Institute of Critical Care and Anesthesiology, Vedanta, the Medicity, Gurgaon. He's the president of Sepsis Forum of India. And he's the president of the Simulation Society of India, had more than 58 chapters in books, 366 publications in several books in critical care, textbook of critical care, atlas of critical care, MCQ in critical care, etc., and of national and international repute. Dr. Arindam Kar, who is now associated with Sir H.N. Reliance Foundation Hospital, Mumbai, as the additional director, critical care, and he was the former director and HOD of CK Birla Group of Hospitals, Kolkata, former director of MICC Medica Super Specialty Hospital, Kolkata, and he is also having 25 plus research publications of national and international. Another faculty who is the director and head of Department of Critical Care at Fortis Escort Heart Institute, New Delhi. In charge of ICU at Fortis, New Delhi. Heads 55 ICU with team of 16 doctors, member of Executive Committee of ICCM Delhi chapter. Teacher of DNB, Critical Care and ICCM courses, in charge of ECMO program at PHI and head of COVID task force in the hospital. Dr. Hiten Karelia is a consultant ID, director, Prime Infectious Care Hospital, Vadodara. and he is the member of Infectious Disease Society of America, IDSA, advisory member of Asia Pacific, Adolescent Health Hong Kong, and having many publications in national and international repute. Doctor, you know that the sepsis is a subject which has always evolved as a subject in critical care and in life-saving management. And there are new therapies which is evolving. And if you remember, during COVID time, we as a company, as a organization, has got the first approval of DCGI, Sepsivac, which is a novel product for the first time in India and in the world. And we saved millions of patients with that. And the approval of DCGI has come for sepsis, and we are the only house who are having this approval. You will be very happy to know that this same product we are applying for US FDA approval from our onco background, and it is going to get the approval within few months with the international approval. And as you know, immunomodulation is a therapy which is now evolving in many of the diseases, and sepsis is one of them. I would like to request Dr. panelists to have your deliberation. After Dr. Yatin Mehta will take you through on the sepsis management and role of Sepsivac, and there is a group discussions and all faculty members will join there. So I hand over this session to Dr. Yatin Mehta. Dr. Preeti will be coordinating now. And thank you very much. Yes, over to you, Dr. Mehta.
Thank you, Devesh. It's a pleasure to have all of you, renowned panelists. Let me just try to share the screen. Okay, can you see the screen now, Devesh?
Yes.
Okay, wonderful. Okay, so I'm going to talk a bit about Sepsivac to introduce for the uninitiated what it is and in brief, and then we'll follow with the discussion. So basically, what we have, if you go by the sepsis 3 definition, is a dysregulated host response leading to organ failure because of sepsis. And dysregulated, what we see in sepsis is there is a inflammatory and anti-inflammatory, pro-inflammatory and anti-inflammatory cytokines which are released, and they are dysregulated, because of which patient can go into vasoplegic shock or can go into inappropriate response to infection, maybe in the initial stages or later stages, which leads to organ failure and vasoplegic shock. So this is how the disease progresses. You'll have pro-inflammatory cytokines released, which will produce vasoplegia, organ dysfunction, capillary leak, activation of inflammatory cells, and so on. So the imbalance is what leads the patient to either, if imbalance is too much, it will lead to organ failure and death. And if it restores, then the patient will recover from it. We have standard current treatment. We have source control, diagnosis, appropriate antibiotics, timely antibiotics, and then you provide organ support like hemodynamic support with vasopressors and the resuscitation fluids, steroids, and then you come to the specific organ support like renal replacement therapy and ventilatory support. Then there are other supportive therapies like immunoglobulins, when to administer blood products, restricted versus liberal strategy, what sort of sedation and muscle relaxant you should give, adequate sugar control, when not to use backup, DVT prophylaxis, test ulcer prophylaxis, and adequate nutrition to the patient. So these are all supportive care. But despite all this, the mortality still remains very high in sepsis. And there are newer therapies being investigated, like activated protein C went and now is dead. ulinistatin, IL receptor like anakinra. Then you have COX inhibitors and bradykinin antagonists, antithrombin 3, thrombomodulin, GM-CSF, and so on. But there are certain limitations to all these therapies. Most of them are unproven of unproven benefit. Ulinistatin acts against IL-1 and IL-6 and TNF alpha, but really the papers are a bit limited. There are not enough randomized control trial. Activated protein C, as I said, it has been not being used now. COX inhibitors have side effects, like renal failure and bleeding diathesis, and other agents have not really been proven. So most of these are not really recommended in the surviving sepsis guidelines. And as I said, the mortality does remain high despite the therapies which we are standard of care. So therefore, that is where the role of adjuvant therapies come into play, and Sepsivac is one of them. What I'm happy about it is a totally indigenous product. It's basically Mycobacterium indicus preni, which is also called in short form Mycobacterium W. It's a non-pathogenic saprophytic mycobacterium, so it does not produce any disease as such. And it's a very potent immunomodulator with multiple sites of action, which I'll come to that. So what does it basically do? It's a potent TLR2 agonist. It's a poly-TLR antagonist, cytokine suppression, gene profiling, and so on. The dosage which is administered is 0.3 ml intradermally in 3 divided doses at 3 different sites. Deltoid is the commonest one which is given, or thigh, lateral thigh, but it has to be given intradermally. That is the crux. So you have to train the staff to do it, because if you inject subcutaneously, other complications can happen, and you have to give it for 3 consecutive days. And this is how the bleb is formed when you give it intradermally. And it, as Mr. Devesh said, it has been approved by DCGI. It was first used in during the COVID times, but it is being used in other studies also. So I'll just a couple of papers which I'll talk about. Let's look at the clinical evidence. So in this paper, patient of sepsis, they used Sepsivac, and they found significant reduction in ICU and hospital length of stay, days on mechanical ventilation, reduction in SOFA scores, and secondary bacterial infection, significantly less in this group compared to the control group. Now, this was a paper, phase 2B study from PGI Chandigarh, and it was a randomized control trial of about almost 200 patients. Yeah. So they had equal number of 101 patients in both group, control arm and the treatment arm, and there was a 50% benefit. Actually, the number needed to treat was 9. And if you remember from people of my generation who used activated protein C, the number needed to treat was 16. This is only 9. So what did they find? They gave it 0.3 ml, as I said, for 3 days intradermally, and there was the control is orange and Sepsivac is green, as you can see. Ventilator free days was less, vasopressor days were less, ICU stays was significantly less, hospital stay was significantly shorter, secondary infection was again significantly less, and ventilator associated VAP was again significantly reduced. So this was good number of patients, randomized control trial from a respectable teaching institute, and very positive study. And again, this SOFA score, as you can see, control versus the study group, significant improvement in SOFA scores. These all, and both at for 14 and 21 days, significant improvement. So they concluded from this study, the Sepsivac was associated with 28-day mortality reduction, time to discharge was reduced, time to discontinuation of vasopressor treatment also was less, and mean SOFA score was significantly reduced. As I mentioned it earlier, it has to be administered intradermally because there are complications if you inject it subcutaneously. So now this is a little tricky. It is a little inconvenient because 3 sites of 0.1 ml each it's not very comfortable. And you have to have trained staff. Intradermal injection is technically a little more difficult to administer than a sub-Q injection. So this was the safety and efficacy of injection Sepsivac. They gave it 0.6 ml in diluted 100 ml saline intravenously. Because all our patients in the ICU who require adjuvant therapies would have an IV cannula. And not only was it safe, they found a significant improvement in all the vitals was observed from in the patients who received the therapy. And this was a study which we did of safety of 0.3 ml IV for 3 days of Sepsivac, published in Lung India. And again, we found no side effects because of the intravenous administration, and we perfectly safe to give. So it is much more convenient to administer. So what are the solutions offered by Sepsivac? It acts pretty fast. So when you normally talk about vaccine, it takes several days or weeks to be effective. This action begins within 2 hours. It improves the natural killer cells and macrophage activity. It restores the CD4 to 8 ratio, generates strong T helper cells response. In sepsis, this has been found to reduce. Cytokine release is again attenuated. Macrophage activation happens because of TLL4 antagonism. Now, TLL4 binds to LPS to initiate the cytokine storm. So by antagonism of this, it reduces the cytokine storm. T cell proliferation happens. Poly-TLR antagonism, the cytokine response is further reduced. It has no side effects, and it also modulates the genetic modulation produces. So Sepsivac can be used in emergency. Timely intervention is important, so it has to be administered in the beginning of the disease as soon as the patient is under your care, because earlier you start, the better it is. Intravenously, 0.6 ml has been found to be better than 0.3 ml. So that is what one should be administering. And obviously intravenous is perfectly safe, so that is preferable to intradermal, I would say. Mortality is a significant reduction, twice the reduction if timely you begin the thing. For intradermal, the dose is 0.3 ml per day for 3 days, and for intravenous, 0.6 ml per day is better than 0.3 ml. So thank you very much for your attention. And if I have, Preeti, if I can have the...
Yes, sir. Thank you so much, sir, for the wonderful presentation. It was very, the insights was very good. So let's move forward to the panel discussion. I'll share my screen.
Sure.
So welcome to our panelist, Dr. Arindam Kar sir, Dr. Rajat Agrawal sir, and Dr. Hiten Karelia sir. Is it visible, sir?
No. Yeah, now it is visible.
Yeah, it is.
Yeah, so let us begin with the senior most guy in the in the panel. So Arindam, you qualify for that. What are adjuvants? And is there a need for adjuvants in sepsis therapy? Arindam there?
Sir, yeah. Sorry. Sir, technically speaking, whichever adds up to an effect, it could be an adjuvant, right? So as you showed very clearly that the mainstay is resuscitation, organ support, antibiotics, and source control. And if we try to do something on where we can actually, because sir, clearly you had been part of the Surviving Sepsis campaign, which has defined sepsis in a newer way, and which has very clearly included the term dysregulation of the immune function. So this part has to be addressed in some way. And most of the adjuvant therapies have been targeted towards taking a control into the dysregulated immune response itself, which is at times, I think, is more important than the inciting factor of infective agent itself. So adjuvants are mostly which are beyond the standard therapy that we are doing, and mostly targeted towards controlling the immune response.
Yeah, thank you. Next, may I have the next slide, Preeti? Next slide.
Yes, sir. Yes, sir.
It's not moving. Yeah.
Yeah, now
Is
it
it?
is.
Okay.
So properties of Sepsivac. Arindam, would you like to comment again quickly?
Sir, mostly as we are aware that this is a saprophytic and non-pathogenic organism. So, sorry, non-pathogenic organism, and most of the vaccine preparations, as far as my knowledge goes, are when it is heat killed or completely killed. So normally it doesn't produce any pathogenic response. Rather, it doesn't have that pathogenic response, but it retains the most of the immunogenic properties itself, which can stimulate the immune response in a more favorable way. And the property wise, it was initially designed to be used as an intradermal agent, but as including yourself, sir, many of the people have used it in a IV format, right? Obviously diluted over 100 mils of saline and given over at least 15 minutes slow infusion. So it has not showed any complications out of it, and it has also shown to be equally immunogenic or equally producing the desired immune response that we have looked in. So as a standard, I would say it is a good improvisation for an ICU patient, because giving intradermal injections in a severely sepsis patient with a compromised circulation and all others. So there are two possibilities. One is it could be a delayed response, and there could be other problems in related to intradermal. And not much of our, honestly speaking, the people who administer, the doctors and nurses, are not very trained in intradermal injections itself. So it is easier for us to go for a IV preparation or IV way to introduce the vaccine.
Yeah. May I have the next slide, please?
Yes, sir.
Preeti?
Before moving forward, sir, I would like to tell the audience. So, doctor, you can also question. You can send the question in the comment box also. At the end, we can discuss later.
Yeah, sure.
Yes. The next question.
So, Rajat, yeah, what are the effects on lab and immunological markers?
Yes, sir. So I think, sir, you have already mentioned some of that in your presentation. So basically, as you mentioned, the dysregulation, which is the crux of current sepsis pathophysiology and current scope of research. So it basically acts on the toll-like receptors, TLRs, which are on white blood cells, and it causes the both pro-inflammation. It suppresses the pro-inflammatory response as well as the anti-inflammatory response. It boosts them. So the imbalance which is there, which is leading to dysregulation, it suppresses that and makes it more effective for the combating the septic shock. On the other parameters are obviously, yes, like cytokines like interleukins, IL-1, IL-6, which are harmful mediators of inflammation and which cause mainly end organ dysfunction in septic shock. It also suppresses their activities. So yes, in that way, it is quite effective in that terms.
Okay, let's have a look. Next one, please.
Yes, sir.
So any drug effects or side effects or contraindications?
Sir, what I have used so far in my experience, I have not seen any major side effect of this medication, of this drug, because it acts well at different routes. So there's no drug interaction has been reported and has been observed in the clinical practice and whatever literature has been published so far. The contraindication would be not be only side effect would be as discussed by Dr. Arindam and you, so that intradermally, when it is sometimes used, it causes blebs and some skin necrosis. So that sometimes is a thing which is observed and reported in the literature. But otherwise, as such, it is quite safe and easy to use.
Yes.
Yeah.
To both of you, just I was wondering on that part, because this is an sort of immune modulator, as we have seen. So any experience or any, I would say, a logical conclusion that if we combine certain immunomodulators with Sepsivac, what could be the plausible effect? Good, bad, contradiction, or it could be as a sort of both can act together, sort of additive effect, synergism, or something. So we have used steroids, we have used a lot of, you have used filters, steroids, other sort of immunomodulators. So what it could be, sir? Any experience on that? I think Rajat, Hitan, you guys can also contribute on.
No, that's a very good question because that is relatively unexplored. Although I have used Sepsivac along with cytokine absorption system in a few patients. Okay, cytosol filter I've used both together, and I've not had any problems. If you use it theoretically, even with ulinastatin, which reduces interleukin 1, interleukin 6, and TNF alpha levels, then I don't think that should be a problem. Although with heme adsorbers, it will reduce all the cytokines, including anti-inflammatory cytokines also. Well, this increases the level of IL-2, which may be beneficial to the patient. So I really can't comment because there are no publications, there is no work on this. But that's a very thought-provoking question. Rajat, any comments or Hitan, anything for you to say?
Yes, we have we have used a couple of patients where patient was in like sepsis and the patient was already on some steroid. At that time, I think we get a better effect because it has some kind of synergistic effect with the steroid, which was a hydrocortisone like 50 milligram 8 hourly, which we are used to give in some septic patients. So at that in that cases where I have been called and when I've used this Sepsivac, and I got a good response. So yeah, probably it is not doing any harm to the patient when it is used in combination with steroids.
I suppose,
yes, sir.
Yeah, Rajat, please. Yeah.
Yes, sir. Sorry, sir. We have used, sir, in patients. All our patients have been on steroids. So we have used in all our patients. Majority of patients were on steroids earlier because, as per the surviving sepsis guidelines, if there's any shock on 1 or 2 vasopressors, they are already on steroids. So we have used almost all our patients already on steroids. Probably combining as the panelists are suggesting with unilastatin or like cytosorb, etc. Probably maybe in vitro studies, if can be done earlier and to see exactly the levels of all cytokines, interleukins, and then we can extrapolate it to the clinical indicator. That would be my humble submission that if we can do that, then it will be great.
No, that's perfectly correct because see, I also used it in the patients. All of them have been on steroids because if the patient is that sick, he would be on septic dose of steroids, which is 200 milligram per day. And obviously, all patients have been on that dose of steroids. So I think steroids are given. Rest of the other things need to be investigated, as Rajat, you rightly pointed it out. Okay, let's have the next slide, please.
Yes, sir.
Okay, so clinical use of MW in sepsis patients, when to initiate, what is the right time to administer? I'll open it to all of you, please, one by one.
Yeah.
Arindam?
Sir, I would ask Rajat to initiate because he has done a major work on it.
Yeah, sure.
Then I will take that.
Right.
Okay. So initially when I started using in 2021, that was a COVID. It was at its peak, and what the literature I went through was that it was used as an intradermal route. Now, at that time, at the COVID, we were very reluctant because nurses were reluctant to give intradermally to a COVID patients. So we thought, and there were some studies about the safety of use in intravenous route 5 ml. I think there were a couple of studies in optic neuritis, multiple sclerosis, etc., where they had found 5 ml was quite effective and quite safe. So we thought, let's start with and there were publications from Dr. Mehta was doing a study and publication in chest as well. So we started using 0.3 ml, not in COVID and in septic shock patients. And we found absolutely it was safe. Though it was a very small study, a small number, only of 30 patients. But yes, what we could found initially was that there was a great significant reduction in the vasopressors from 40 to 72 hours onwards. Though we could not extrapolate it to the mortality benefit because it was a single arm and the number was quite small, but it was absolutely safe. And another important thing was the majority of patients were elderly. So, we get a slightly geriatric population age group in our hospital. So majority was 65, 70, and there was no side effect, and all our patients were in septic shock. So this was the experience we had initially.
Yeah, also see, I also, after my study was over, I also started now using it intravenously. Why to give 3 shots to the patient? COVID times you got away because the patients mostly were that sick and intubated, so they didn't know what injections they were giving. But if the patient is not that sick and is awake, I think 3 intradermal injections is not a very great thing to do and a memorable experience for the patients to give. So intravenous is perfectly safe to give. Also, as showed in a few studies, 0.6 ml is better intravenously than 0.3 ml. That means double the dose what we do intradermally. Fortunately, it's not a very expensive drug. And also earlier you probably give it, the better it is. So don't wait for the patient to progress to septic shock. If the patient is already with you, then it is better to start early before the organ dysfunction starts. Like any adjuvant therapy, I suppose. Waiting for too long, maybe once a patient goes into multi-organ failure, none of them will be salvageable, whether you give Sepsivac or you do not give Sepsivac. Hiten, anything to add?
Yes, and actually you correctly mentioned everything. What I feel that before, you correctly said before, in myopeties, when the patient having a single organ failure and the patient is in sepsis, then at that point of time, I think I am already late. So I would like to start this Sepsivac as early as possible after the the initial antibiotic has been given within 24 hours. And yes, I also prefer nowadays IV route, because many times intensivists were asking me that why you are giving IV when it's mentioned that it's intradermal. So it was quite resistance in our setup. Then finally we convinced to give in the form of saline. And yes, we are having a kind of small data, which is, we are just making it refined. So yes, I agree with you, sir.
One quick
Sir,
question to the,
Oh, sorry, sir. Now you carry on, then I will.
One quick question I had for the Cadila team. Is it DCGI approved for intravenous administration now?
No, sir. No, sir. It is DCGI approved for the intradermal route, not for the intravenous route.
So you should apply for it, no? On the basis of a
Yes,
few published studies, you should now apply for it. Yeah.
Yes, sir. Definitely, sir.
You can give Rajat's name as an expert if he for the information. Yeah. Okay.
Sure.
Yeah,
Sir.
Arindam.
Now, I was going to point this out. Actually, do we need to be, because all of our are working in most reputed institutes. So do we need some ethical clearance to use vaccines in a non-permitted, if I say so, which is clinically safe, right, proven, but not being permitted by DCGI? I think sir has given you, Prithiji, the guidance actually. So as soon as possible, let us take that. It would be easier for most of the clinicians to use in
Yes,
that
sir.
way.
Definitely,
Sir,
sir.
I was intrigued on one point that given in the intravenous route, why do we need to double up the dose? What could be the plausible explanation on that part? So for a quicker response, for a more magnified response, what it could be, sir?
That's an interesting question, because you're right. Intravenous, normally the drug levels are much higher than intradermal administration. I don't know, but that's a published study. 0.6 ml was better than 0.3 ml.
And secondly,
comments?
sir,
Yeah.
Oh, sorry, please.
No.
No, you asked Rajat, I think.
Yes, sir. Sir, what we found was, sir, that once we used 0.6 ml, first of all, it was pretty safe. There was no side effect. Secondly, again, we did not have a control arm with either 0.3 or with intradermal route. So then we could have seriously compared and then exactly knew how much difference is there, whether it's clinically significant or not. But what we could see was initially, though it's too presumptive to say the response was better. And yes, if the response was better before the onset of renal failure, like initially we were a little hesitant and skeptical in using it. So we use it as just as an add-on therapy as a last resort. We did not get much results, but before the onset of renal failure, when we started using IV 0.6, the result was slightly better. But again, no p-value was calculated. Again, it was just a clinical experience which was there. So we don't have that randomized arm to prove that really.
And then I'm sure Cadila was not too unhappy with the study because you're using double the amount of what is marketed at. Yeah, Arindam, second question.
Yes. Sir, the other thing is that the timing, right? So as you pointed that most of the times we think of any of the adjuvant therapies as a salvage, which I think had been put a lot of products or interventions into disrepute, because if your timing is not right, if you are not early, then it is of no use, I would rather say. And the other dilemma is that if you are using it too earlier, right, it may not be appropriate in a milder cases. So can there be something as we are ongoing a trial with one of the hemoadsorbents where we are measuring certain immune markers and accordingly timing our therapy on that part. Sir, can something of this could be thought of? Because now all the immune markers are quite measurable, and it is at least in the major metropolitan cities, those markers are actually being measured by most of the labs. So can we think of something where we can have a quantifiable immune marker in our hand, and then we can say, no, this patient has a quite a inappropriate pro-inflammatory response to the sepsis itself, and then we should be justified in using most of the adjuvants?
No, that's a good question. I think that needs to be explored. There are no published studies on this. But like for hemoadsorber, most of the German studies have a cutoff of 500 of IL-6 levels. If it's exceeding 500 micrograms, then there is significant pro-inflammatory cytokine response, and that is the time to sort of intervene. So that's again a food for thought for the Cadila team here. I think they have to support certain studies to be scientific studies, academic studies, to come out with more evidence in favor of that.
Yes, sir. Sure, sir.
Now the next.
Yes, sir.
Next question.
Next question.
Well, this we have already discussed. I think
yes,
the IV route,
Yes,
again, I will say, I think at the moment, till they get a DCGI permission, it is safer to get a consent from the patient. And I'm sure the DCGI should not be a problem. There are enough now published studies to show the efficacy and safety. So I think once that comes through, then I would recommend IV routes. Although I have started using IV, I do take consent for the different route for the patient. But IV is much simpler to give. You're not dependent too much on the expertise of the nurse who's administering it, and less painful to the patient who's not necessarily on a ventilator and sedated. So I think it is better.
Okay, sir.
Any comment, Rajat? I think Rajat is the last study.
Yes.
So published study.
Yes, sir. Sir, I think yes. As sir you mentioned, we are using IV now and 0.6 ml. The duration is 3 days. What we are using 0.6 ml, we have used IV for 3 days. Though it might be considered used in a long term, but the exact duration still probably needs to be fixed and defined. Probably once we have more randomized trial, we can have how long it can be used. But so far we have used it for 3 days intravenously.
No, at the moment that is what the recommendation is, for 3 days. So I think till something new comes up, that is what we should stick to. Yeah. Next one, please.
Yes, sir.
Safety concerns, anybody? Hiten?
So I think, sir, being a kind of vaccine and intradermal route, apart from this, local kind of blister and anything, it doesn't have any safety concern. And obviously, we are definitely going to use some associated antibiotic, which are quite safer. and having I have not found any kind of literature which is saying it has any drug-drug interactions. So I think it's pretty safe.
Yeah, see, the only complications which have been reported are basically if they're not injected inappropriately or subcutaneously, then they can have skin ulceration and a local lymphadenitis. So I think that is what one has to be careful. And really contraindication if the patient has got exfoliated dermatitis or has got eczema all over the body or has got pustules, then obviously you will avoid giving it those sites. But otherwise, there is no real contraindication. And every new product, the manufacturer always mentions that not recommended in lactating women and pregnant women, because obviously studies are not done in that and safety issues are not proven. So don't use it in lactating and pregnant women. And they also always mention anybody who's a known allergy. So if somebody is shown in the past, but that's very, very unlikely. This is a new product and unlikely that that patient in whom you use Sepsivac will come back after a few months again who requires a second dose of Sepsivac. So it's most unlikely. So I don't think that is really applicable. Next one, please.
Yes, sir.
Sir, I have one question.
Yeah.
Raised hand. Sorry, I'm always putting up the question. You know, the quintessential argumentative Bengalis will always come up with questions.
Arindam, I didn't say
You
it.
have lived with it.
I lived with one, yeah.
So you know that. Sir, never mind. Two things. One is that I think the people the patient whom do you choose are eligible for Sepsivac, almost all of them would have a central line. So should we put it in that way that it would be easier or it would be sort of lesser complicated if we go through the central line? Can we sort of discuss about that, sir?
No,
or is there any?
No, I don't think so. I don't think so there's any problem giving it in a peripheral line. So why, I mean, insist on the central line? I mean, that will only
I'm not insisting. I'm saying it is maybe more
no, why?
sort of
it
easier
makes no
advice.
Difference.
Okay, sir. So it's not mandatory. So we can give through any of the peripheral or even central line, right?
You can give it any peripheral line. So you can see the main thing, what it changes is, since you can give it in a peripheral line, you can start it in the emergency if you want to do it.
Exactly. So
that is maybe the advantage.
It expedites the entire process.
Yeah.
Otherwise, if we make it that go through the central line, then people will wait and an additional time would be lost, sort of thing.
Correct.
Secondly, sir, India has quite a bit of the load of Mycobacterium, right? The standard Mycobacterium and others.
Yeah.
So I think it's to all of you, including obviously led by sir. So do you think that the response could be altered in a patient who is already infected with Mycobacterium?
No, I don't think so. Because what the literature says is, even there is no interaction with anti-tubercular drugs or anti-leprosy drugs, and there is no issue with that. Because it's a different Mycobacterium. It's not the Mycobacterium which we are used to. So, any comments on that?
Yes, I think it has a multiple antigens. So not only this routine tuberculosis antigen, but it has a multiple antigens, so it might be helpful too.
In fact, I think what I believe is that once this molecule, the heat-killed Mycobacterium pranae, was initially studied for Mycobacterium leprae as an anti-leprotic therapy. So, and this is a heat-killed. So, what I know from my virology, Dr. Hiten can be better to answer that. But what I know, I don't think it has any. It promotes the effect of Mycobacterium tuberculosis or anything like that. So in that way, yeah, it is there.
So there is no problem there. Yeah. Next one, please.
Yes. This one is the question.
How to monitor the response? Anybody?
Sir, we use only the clinical parameters mainly, like the SOFA scores. And the another thing which we are now started monitoring is, which we are not doing it earlier, is the lactate clearance. Though it indirectly reflects the reduction of vasopressors and resolution of end organ damage. But yes, that also we are monitoring apart from the clinical response.
And I suppose your white count will be coming down, your CRP levels will be coming down. So those are things which we can look at. But otherwise, it's mostly clinical that your organ functions will get better and your severity scores will come down, vasopressor requirement will come down. So those are the things which we need to monitor. At the moment, there are no other biomarkers which will really guide us whether it is effective or not effective. Next one, please.
Yes, sir.
Open to all of us. What's the background diagnosis of sepsis? Etiology of sepsis?
Most of the studies and whatever personally we all have used is your standard causes of sepsis and obviously
Bacterial
the bacterial sepsis and respiratory system being the most affected one, followed by the intra-abdominal sepsis itself. So I think these are the two, and most of the times with our experience that we have seen the more hyperimmune response are common in community acquired infections, right? Because they are more virulent and more dramatic on that part. I think so. This is the trend that bacterial sepsis, respiratory tract, and mostly community acquired. These are the three areas where maximally the Sepsivac has been used and with good result.
Rajat?
Yes, sir. I think I agree with Dr. Arindam. Yes, we have used in pneumonias and intra-abdominal sepsis. Presumptively gram-negative, but then yes, it was not 100% culture proven. So yes, so these were the two main sources of that is pneumonias and intra-abdominal sepsis. We did not use in fungal sepsis as such. We did not use it in that, but only bacterial sepsis.
Yeah, I think all of us have used primarily for bacterial sepsis. I've used it for cellulitis also, urosepsis also. Few patients I've used it for. So I don't think, I mean, as long as it's bacterial sepsis, you are okay. There's no real preference that it has to work only in the respiratory or it has to work in only bloodstream infections. I think it'll be quite effective in any system involvement. Yeah, next one, please.
Yes, sir.
This has already been
yes,
answered, I think. Do we have any questions from the audience, Preeti?
Yes, sir.
Okay, let's first look at this. Therapeutic vaccine, sepsis and emergency. How can vaccine be so far? This we already answered. I think within 2 hours, you'll start seeing if you're measuring certain biomarkers, then 2 hours, you should start seeing the effect of that. Not only the cytokine response, but also you'll see macrophage activity increasing and killer cell activity increasing and all that. So it has been measured. I mean, the effect starts within 2 hours, unlike all the other vaccines.
And also, sir, theoretically, most of the vaccines acts as producing or generating specific antibody against the organism itself, wherein this is a generalized vaccine which is actually promoting certain and anti-inflammatory effects and reducing the pro-inflammatory part. So it is not creating any specific antibody or building up to it. So that's why it is much faster to act upon, and it initiates the response very quickly.
And it has a very extensive immunological response. It affects so many different cellular functions. It's not a specific anti-cytokine, but if you look at the activity, macrophage activity, apoptosis decreases, and multiple and agonist of TLR2 and antagonist of poly-TLR antagonist. The T helper cell activity increases, CD4 CD8 ratio is restored to normal. So there are multiple effects of the medication. Next one, please.
Sir, here we have ended the panel discussion. So there's a question from the audience, from the Dr. Johnny from Amritsar. Sir, he's asking that what is the role of Sepsivac in the gram-positive and fungal infections? Sir?
First, I think I'll Arindam, Rajat, any comments?
Sir, we have used, as we discussed in the panel discussion, we are used mainly in the bacterial sepsis. Though we are using community-acquired infections as well, some of them have been gram-positive, but we have not differentiated between the two. Intra-abdominal infections are predominantly gram-negative. So as such, we have not used in fungal sepsis, but yes, bacterial sepsis, it works quite effectively.
Okay, sir.
See, I don't think it is any preference for a gram-negative or gram-positive bacteria, although most of the studies are gram-negative sepsis, because it's an Indian invention or discovery, whatever you call it, invention. And therefore, gram-negative sepsis is more common. So that is why it is like that. I have used it in cellulitis, so it should be effective against gram-positive. But primarily, it is in a bacterial sepsis, it will be effective. I don't really know how effective it will be in fungal sepsis because there's more of a cellular immunity and all that are involved. So we really can't comment on that.
I think the question is also quite valid. In there are certain differences when we are dealing with gram positive, particularly very virulent community gram positives with where they have exotoxins and others, particularly the CMRSA or something of that sort. We have had quite a good experience with the IVIG, which acts as an antitoxin itself, and it would be clearly in the subsequent trials in Sepsivac where we can really differentiate into gram positive and negative. I think Rajat, you can lead there. I think we can have a much clearer answer. But normally there are some certain final common pathways where the harm is being done, and Sepsivac really do work on that part. So it should be applied for gram positive also, but maybe we have to use certain additional things in the gram positives. That's my viewpoint.
Yeah, that's fine. I think we should use it for bacterial sepsis. It is unproven in fungal and viral sepsis, although it has been used quite extensively in COVID times. We have used it quite a lot, and so has Rajat. So, yeah.
Okay.
Any other question?
So one last question is it effective in later stage of sepsis? The question is from Dr. Sanjeev Kumar from Patna.
Yeah, Sanjeev, yeah. So, well, we really don't know. If the patient is in multi-organ failure, if you look at with Apache more than 25 or 30, the possibility of his dying is pretty 60-70%. So whether it will be effective or not effective, it is very difficult to comment at that point of time. I see no harm in giving it, but again, you may lose the battle. So I mean, why to try a relatively new drug in a patient who's almost dying? So I won't recommend it very late stage. Yeah.
Okay.
That's also create a problem on that. So then you start blaming that it won't work. It doesn't work, right? If you're too late on that day, because by that time the fate has been decided, and if you are trying to, as I told in the beginning, that you try to as a heroic or salvage therapy is not going to work on that. I think Sir has very clearly shown that in the first 48 hours, even before the first organ dysfunction, or maybe the first organ dysfunction has already started, because most of your patient would be hemodynamically unstable by definition in a sepsis or septic shock. I think that is the best time to initiate, to take early control of the immune dysfunction itself, rather than waiting for the day. And also at the end stage, maybe the process overall, the fight between pro and anti, and then process is little different from what we see in the initial period, the TLR4 mediated and others. So I think it should work in the early phases, not in the late phases, even in the theoretical framework.
So I think we had a good discussion about this relatively new, indigenously developed so-called vaccine, but which has significant immunomodulatory effect, and which has had several positive studies. The only limitation was the intradermal injection, which has been taken care of by a few papers, that can be safely given intravenously. It has really no side effects. It's not a very expensive drug, and I see no reason why not to give it as an adjuvant therapy when you're given the standard of care. So I think with those words, I would like to hand over the proceedings to the Cadila team again. Thank you very much.
So thank you very much, all panelists, Dr. Mehta. I was looking at the watch list. Actually, there are around 450 doctors across the country, along with Nepal sir was watching your deliberation and your group discussion. So the number which I have received just now, it was 450. Will be actually having more number from Nepal also. So it will cross 500. So I feel, sir, it was a very, very good discussion. I request Arnav, who is our Senior General Manager, to have a quick video of the corporate, and then vote of thanks will be given by Sandeep Mohan, who is our Cluster Head and Vice President. Over to you, Arnav.
Sir, thank you. Thank you. Thanks, Mehta sir, Dr. Arindam Kaur, sir, Dr. Rajat Agrawal, and Dr. Hiten Karelia, sir. Thank you. Thanks for all of your participation. And in coming days also, with the leadership of Devesh sir, we would like to continue this kind of initiative on a regular basis, which will create a good amount of presence in the critical care fraternity Cadila as a company.
Hello.
Because we as a
Good
company,
evening.
we would like to inform you that
Yeah, I'm just joining
along
in.
with our Dholka factory, which right now we do have, which is having itself USFDA approved facility. Along with that, our management has decided, and from coming July of 2024, from the first time in India, Cadila will provide you the small volume parental item, which is getting manufactured in a USFDA approved plant. So to initiate this further, Cadila as a company, Voltacare as a division, we would like to do this kind of knowledge dissemination program. We need all of your support. We need all of your guidance, which will take us further forward. Thank you. Thanks once again from my end, from Voltacare team, Voltacare end. Thank you for all of your participation. I would like to request Dr. Preeti, please do share this corporate presentation for the group, and then Sandeep sir is here.
Yes, sir. Sure, sir.
Behold, the story of victory, of life over malady. Behold the saga of power, of science over sickness. Behold the tale of triumph, of dedication over disease. Behold the rise and rise of Cadila Pharmaceuticals Limited, one of India's largest privately held pharmaceutical companies based out of Ahmedabad, Gujarat, India. Cadila Pharmaceuticals was born out of the vision of a legend who left an indelible mark in the chronicles of Indian pharmaceuticals industry. He was Indravadan Ambalal Modi, the founder chairman of the company, rightfully christened as the medicine man of India. For Sri Modi, healthcare was not a privilege of the few. It's the fundamental right of all.
In 1951, I know that when pharmaceutical industry was not existing in this country, and people felt that being a technology-oriented industry, perhaps it cannot be started in this one. And at that time, I decided that in the interest of the country and everything, better we may give a foundation so that other people also follow for this industry.
His vision to alleviate pain of the underprivileged through research-driven innovations has been driving Cadila Pharmaceuticals for more than 6 decades now. Innovations which are backed by the key strengths of the company, the state-of-the-art manufacturing and research facilities. The flagship manufacturing facility at Dholka, Gujarat, sprawls over 100 acres. This avant-garde facility is USFDA approved, besides meeting most of the stringent quality standards across the globe. It manufactures tablets, capsules, injectables, liquids, and oils. The second formulations manufacturing facility is located at Samba in the state of Jammu and Kashmir. The API facilities at Ankleshwar, Gujarat, manufacture a large number of key APIs and intermediates at this USFDA and ISO 9002 and ISO 14001 accredited plant. The company also has set up its first ever overseas formulation manufacturing facility at Addis Ababa in Ethiopia. This world-class facility is the only WHO cGMP compliant manufacturing facility in the region. Cadila Pharmaceuticals offers an ensemble of formulations. The company's extensive range of dosage formulations covers more than 850 products spread across 12 specialties, with many of India's top brands in its basket. Great goals are defined by far-sighted innovations, such as Polycap, the world's first polypill for cardiovascular diseases; Rizorin, the world's first boosted rifampicin for tuberculosis; Mycidax-C, the world's first active immunotherapy for lung cancer; and novel formulations like Rabiloc IV, the world's first rabeprazole in IV form. To take the focus on innovation to the next level, Cadila Pharmaceuticals believes in forging scientific collaborations with world-class institutions sharing the same passion for healthcare research. It partners with leading international companies to discover newer treatments in the areas of novel vaccines, better antibiotics, respiratory diseases, gastrointestinal diseases, cardiovascular ailments, diabetes, cancer, sepsis, and regenerative medicine. The Cadila Pharmaceuticals Group is gigantic. Its group companies complement and expand the dynamic vision of the company across new avenues of healthcare and newer fast-paced business areas. Cadila Pharmaceuticals is a gentle giant. It undertakes a number of targeted activities that are bringing about long-lasting and deep-rooted changes, especially in the fields of health, education, women empowerment, and child care. As a responsible corporate citizen, Cadila undertakes a number of CSR activities in these areas. When the beacon of exemplary leadership passed on from the illustrious Doyen Sri I.A. Modi to Dr. Rajiv Modi, Cadila Pharmaceuticals scripted a new success story in its journey of fast-paced growth phase.
For me, it's a mission, and I have a very simple mission, which is to take this company forward at all costs, to make it into a globally sized business, which will continue to provide the world's best quality, affordable medicines for people around the world, and would strive to bring innovations.
Strategic global expansions and research into areas of unmet medical needs are the future. Decades of dedicated research, groundbreaking healthcare innovations, and worldwide impact proves the infinite potential of one powerful emotion: care. Care that transforms into a driving force for a revolutionary change. Care that translates into affordable miracles for those who need it the most. The success of Cadila Pharmaceuticals bears testimony to the fact that the care continues.
Thanks,
Thank you.
thanks, everyone. Thanks,
Sandeep.
everyone. I think Dr. Mehta's left, but thanks a lot, Dr. Arnab, Dr. Rajat, and Dr. Hiten. I think we are really thankful to you. I think, and what came out today from the meeting was earlier the better. That was the thing which everybody, all the panelists, including Dr. Mehta, said, and that is what will go down to the audience and the respected doctors who are watching this meeting. And this is just a start, as our Arnab Ghosh said, and I'm sure we'll looking forward to many more such meetings, Dr. Arindam, Dr. Hiten, and Dr. Rajat from your side. Your support will be required, and to passing on this knowledge as well as the wonderful molecule which can save lives, as we talked, sir. Thank you very much. Thanks, Dr. Preeti. Thanks, Dr. Das. Thanks, Arnab. And thanks, everyone, once again. Good night and have a safe night.
Thank you so much, everyone.
Thank you.
Thank you.
Thank you, sir. Thank you.
Thanks, sir. Thanks.
Thank you so much.